Okay, so what I would like, and I will try to do today, is to very briefly summarize how the innate and adaptive immune system can face microbial and tumor challenges, how or what are the questions we ask during the past few years, and how these questions have filled the gaps of the literature. And without going into detail into experiments, I will try to summarize some of the messages and results we got during the past years. And finally, I would try to share with you how a project that started in 1993, some years ago, from a very basic research, has ended in biomedical implications and with patents that are now in the terms of being licensed. So I will try to start by summarizing some of the key aspects of the immune response. What is the mission and the role of the immune system? The role of the immune system is to defend against foreign microbes, which includes parasites, fungi, bacteria, and viruses, and to eliminate tumors. And also to promote immune tolerance and homeostasis and to silence the immune response. How does the immune system do that? If I go here, and if we go to the wall, and this wall is full of pins, full of the bacteria that has the Clostridium tetanis and the tetanus toxin, we will get infected. And the first arm of the immune response that will act and will serve to eliminate this pathogen is innate immunity. If we start and go to the books, to the textbooks that we used to read, for example, with Alberto, me, or people from our generation, that we read in the immunological books, so we see as a synonymous that the innate immune system is exactly the same as non-specific and unspecific immune response. We know it's not true. So we now know that innate immune system is highly specific. Very recently, last year, Jules Hoffman, in fact, won the Nobel Prize together with Bruce Butler for identifying the specificity of the innate immunity. They found that there are pattern recognition receptors that specifically recognize patterns that are associated in pathogens and can signal inside of innate immune cells, including neutrophils, macrophages, dendritic cells, to activate the immune response. Another of the winners of the Nobel Prize of 2011 was Ralph Tayman. Ralph Tayman, who died and passed away 2 days after they knew about the Nobel Prize, identified for the first time the dendritic cell. What are the dendritic cells? Dendritic cells are probably the most important innate immune cells in the body. Dendritic cells are at the interface of innate and adaptive immunity. So when a toxin or a pathogen is invading our body, so dendritic cells, they can recognize this pathogen and this antigen using these pattern-associated recognition receptors and can trigger innate immune signals to activate these dendritic cells. These dendritic cells then migrate through lymphatic vessels and they arrive to secondary lymphoid organs, including lymph nodes and including the spleen, to activate T cells. So how do dendritic cells activate T cells? So I would like to apologize with those that are familiar with immunology because I am a little bit reductionist here. So T cells, they arise from the thymus. The thymus is called the T cell school because here T cells, they learn how to recognize foreign peptides and foreign antigens, and they also learn how to avoid self-recognition to eliminate and avoid autoreactive T cells. So then T cells that arise and emigrate from the thymus, naive T cells, they go through the blood circulation into the lymph nodes and they find and they meet the dendritic cells. So how do they meet their lover, the dendritic cells? So Imagine that you are in a pub and you went to dance and you are trying to find the love of your life. So first, in the first 3-4 hours, you go in the pub and you start looking for the love of your life, and you generate this kind of weak interactions with people until you find the love of your life. And when you find the real love of your life, you will start to generate strong interactions and strong kisses and strong signaling. So this is exactly the case. So this is the case where dendritic cells, they meet their right T cells with the T cell receptor. They interact with a very high affinity. In the previous film, I can switch it off. So in the previous film, you could see dendritic cells that are trying to interact with different T cells, but they cannot establish a strong interaction. This is totally different from dendritic cells that establish very strong interactions with T cells. And what they do, then how do dendritic cells activate T cells? When they find the right T cell, there are two specific and important signals. One is highly specific, is the dendritic cells presenting a peptide antigen in the context of a molecule called complex Mayor histocompatibility complex, and the T cell, the naive T cells, recognize specifically this peptide. And a second signal that is called co-stimulatory signal that is mediated by B7, CD80, and CD28. So after activation, these T cells got expanded. So from one cell, we get millions of identical cells that they go to fight against the pathogen that has came and got infected. So not only the interaction between dendritic cells and T cells can activate these T cells, but it can also polarize the immune response. It can also tailor adaptive immune response. So, for example, if dendritic cells interact with T cells, and these dendritic cells are presenting an intravesicular pathogen, such as, for example, tuberculosis, Mycobacterium tuberculosis, so we will see the generation and the differentiation of Th1 cells. Th1 cells are important to eliminate intravesicular pathogens, are important in the case of tumors, and they can also generate autoimmune response. In other cases, dendritic cells can also polarize cells to generate Th2 responses that are important for allergic reactions and to eliminate helminth parasites. Also, very recently appeared Th17 cells, which are important for sustaining anti-immunity and for immunity to fungi and bacteria, and also T helper follicular cells, which are important for immunity against extracellular bacteria or toxins. So this is the case of the toxin that infected our body. Also, very recently, has emerged the idea that dendritic cells not only can activate T cells to generate a universe of effector T cells, but can also stimulate regulatory T cells to silence the immune response. So the interaction between dendritic cells and T cells in the lymph node not only activate different arms of the army of the immune response, but they can also silence the immune response to say, stop, it's enough. You don't have to stimulate again. If you continue stimulating the immune response, then there will be an autoimmune response. So during the past 5-10 years, we have witnessed a revolution in our understanding of the contribution of genomics and proteomics to innate and adaptive immune responses. And we know much less on the contribution of glycomics. Glycomics is the complete set of sugars or glycans that cover the cells and cover the tissues. So one of the possibilities of this discrepancy is probably the technical limitations to analyze glycans and sugars during the immune response, but probably another one is related to the complexity, where in genomics we have 4 different nucleotides and 1 type of bond. In proteomics, we have 20 amino acids to analyze and 1 type of bond. And in glycomics, we have a diversity of different type of bonds and multiple branching. So in contrast to the traditional idea that glycans and sugars decorate the surface of cells, evidence has emerged that the glycome has the potential to display over 100 million more information than the genome. So we now need to understand how this information is decoded, how we can decipher this information. So what is the relevance of the glycosylation in the immune system? Glycosylation is important in almost all the processes of the immune system, in almost all the processes that we describe. Glycosylation is important for thymocyte development, for negative and positive selection. Glycosylation is also very important for T-cell activation and signaling, for T-cell trafficking, and more importantly, the glycosylation of the Fc region of immunoglobulins determines the pro-inflammatory or anti-inflammatory activity of immunoglobulins. So this is very important from the clinical point of view. If we can dissect the glycosylation of endogenous immunoglobulin or intravenous immunoglobulins, we can treat patients. And we can treat patients with immunodeficiencies with pro-inflammatory immunoglobulins and those with autoimmune diseases with anti-inflammatory immunoglobulins. So this paper that was published in Science demonstrates that also translational research can be published in high-impact journals. So just in this month, 2 weeks ago, Immunity published a cover of our work, of our article, showing the main message of our work during all the past years, which is deciphering the glycocode in immunity. Here, if you have a BlackBerry, you can decipher the QR code, and you will be able to see the message of the cover. But the most important message is that the responsibility of deciphering the biological information encoded by the glycome is assigned to endogenous glycan-binding proteins or lectins. Among those glycan-binding proteins or lectins, we have C-type lectins in the immune system, with the most important are DC-sign, which is the receptor for HIV, for example, selectins, which are very important for trafficking, Siglecs, which are very important in B-cell biology, and galectins. We will focus today on galectins. So what are galectins? Galectins are a family of animal lectins that have affinity for N-acetyl-lactosamine units, which is galactose beta-1,4 and acetylglucosamine. There are 15 different galectins, which we now know that each of them interact with the different sugar next to N-acetyl-lactosamine. They are highly conserved throughout animal evolution. So we can find galectin-like proteins in viruses, in bacteria, in parasites, in plants. Also in Arabidopsis thaliana, we have two different galectins. So galectins are found in different species throughout the evolution. And differently from the typical ligand-receptor interaction, galectins can form a type of lattice or network and supramolecular complexes that trigger different signals with avidity. So galectins can bind to a wide array of glycoconjugates, both on the cell surface and extracellular matrix, and deliver intracellular signals to regulate diverse biological processes. So here is a typical classification according to the structure of galectins: prototype, tandem repeat, and chimera type galectins. We will focus today on prototype galectins, which have one carbohydrate recognition domain which can dimerize, and particularly on galectin-1. As you can see here, there is a probability that each galectin can form different types of networks or lattices to trigger different signaling responses. So one of the questions of the literature is how we can predict whether there will be a typical ligand-receptor interaction or a supramolecular complex called lattice that is triggering different signaling pathways. So when did our story start? And I will try to go when I was young or younger. In 1992, as you know, I am from Córdoba, Argentina. Probably you can still listen to my accent from Córdoba. In 1992, I was doing my pre-graduate thesis work in the laboratory of Dr. Carlos Landa, who was in the same lab from Ugo Massioni, in Sikievik, in the Department of Biological Chemistry. And we were working trying to generate polyclonal antibodies against glycoconjugates and lectins isolated from chicken retina. So nothing related to immunology. So at the end of 1992, Carlos Landa decided to leave his academic career. So I decided to look for a place to do my PhD. And I loved immunology. I always say that immunology has saved my life because I love immunology. So I wanted to go to the immunology department. And then in 1994, I found finally I could start my PhD thesis work under the supervision of Dr. Clelia Riera. At that time, I got a project, and I always like to talk about this because it was a very unsuccessful project. I couldn't get any results during 2 years, and I was really frustrated. But during this type of nights, when you don't get results, and you have a clean Western blot with nothing, so I started to think what I will do of my life. And I remember that when I finished my student thesis work, Carlos Landa was very generous, and he told me, you have generated antibody, you can bring with you your antibody, you can take your antibodies. And I put the antibodies in the fridge of my mom's and dad's house. And from then, I didn't touch them. So at this night, I remembered these antibodies, and I called my mom, do you still have the antibodies in the tube? I talked with my professor, that I was doing my PhD thesis work. She allowed me to do experiments. And at the end of 1994, galactins did not exist at that time. They were called beta-galactoside binding lectin. Using one of these antibodies, one of the panel of these antibodies, we found a protein band of 14-KDA that we sequenced. And after the sequence here, due to also to the generosity of people from Buenos Aires, I could sequence the band, and it was a 14-KDA beta-galactoside binding protein that we now know that was the first identification of galectin-1. So it was published in 1996. So what we learned during these years? Galectin-1 is a non-covalent homodimer composed of subunits of 14-KDA. It can modulate the survival of T cells. It is expressed at sites of immune cell activation, such as lymph nodes and spleen, and it's also overexpressed at sites of immune privilege, including the eye, the neurons, the prostate, and most importantly, the placenta. It is considerably upregulated from 10 to 50-fold in different types of tumors and metastatic lesions. More importantly, probably, galectin-1 in vivo can suppress chronic inflammation and autoimmunity and induce a bias toward Th2 response in different models of autoimmune diseases. So we found that when we incubate T cells with galectin-1, this protein, was killing all the T cells that were in the plate. So we reasoned that probably it could be a nice treatment for those diseases that have an increased and exuberant number of T cells. So we first demonstrated this in arthritis, and then we were very happy to see that it was confirmed in different models of hepatitis, inflammatory bowel disease, graft versus host disease, uveitis, and diabetes. So one of the most important challenges for immunologists is related to tumor immunity. So one important question is, if T lymphocytes can recognize transformed antigens on the surface of tumor cells, why do tumors grow and colonize new tissues? If we take a photograph of a tumor that is growing and is doing metastasis, we will see a lot of infiltrate. We will see a lot of T cells surrounding the tumors. But these T cells are paralyzed, are anergic. They cannot kill the tumor. Instead, the tumor have devised a lot of strategies to eliminate these T cells. So this is what we call, the immunologists and tumor biologists call the Great Escape, like very similar to the film The Great Escape. So tumors have devised different strategies to escape the immune response. This is like this. T cells wish to kill the tumor, to eliminate the tumor, but then the tumor tried to kill the T cell. And this is what we call tumor counterattack. So in 2004, we were able to demonstrate, I will not speak in detail about the experiments, but I will only try to leave you the message. In 2004, we demonstrated that tumor cells, while they get metastasis and while they invade new tissues, they release high amounts of galectin-1, which binds to glycoconjugates on activated but not resting T cells, and it can kill by apoptosis these T cells. So this is what we call the sweet kiss of death. So in summary, what we found is that galectin-1 substantially contributes to the immunosuppressive activity of melanoma cells. When we blocked, this is a typical growth of a tumor. When we block galectin-1 expression, we see almost complete reduction of the tumor, of the growth of the tumor that was recovered when we depleted CD4 or CD8 T cells. So blockade of galectin-1 within the tumor microenvironment can result in CD4 and CD8 positive T cell mediated tumor rejection, and silencing galectin-1 gene expression induces markedly increased Th1 responses. So we confirmed these findings in a model of Hodgkin lymphoma, and we found that Hodgkin lymphoma, but not other types of lymphoma, express very high amounts of galectin-1, and this is also very important because it served for another patent, because we could demonstrate that there is a differential diagnosis between Hodgkin and non-Hodgkin lymphoma based on galectin-1 expression that biochemists are trying to implement now. So we were very happy to see that this year, this effect, this tumor escape mechanism, was confirmed in lung cancer, in pancreatic cancer, and in neuroblastoma, saying that galectin-1 is expressed by tumors to evade the immune response. So another big question is immunology, not only in immunology, but in biology in general, is why doesn't your mother reject you? So from psychological point of view, all our mothers will never reject us, will say that you are thin, you are nice, you are beautiful. But in the case of immunology, there is a paradox. If you are boyfriends, probably. No? No, okay. So probably you will be boyfriends then, after this meeting. So if you are boyfriends and we transplant some part of the skin of him to her, she will reject the skin because they have different histocompatibility genes. But in the case that they want to have a baby, and I don't want to predict the future, but in the case that you want to have a baby, her baby, the fetus that is growing, will have half of paternal antigens and half of maternal antigens. And in most cases, she will not reject the fetus that is growing. So the most important question is why? Why the mother doesn't reject the antigens of the father in a semi-allograft fetus that is growing. So Peter Medawar, many years ago, he tried to analyze the concept of anatomical and mechanic phases, but this is not the case. We now know that there are a lot of many active mechanisms to inhibit and suppress the immune response that are triggered and propagated from the placenta to the immune system. So in this case, what we found, and I will try to make a long story brief, we found that galectin-1 knockout mice, so mice that do not have galectin-1, have increased fetal loss, and this effect can be rescued by addition and administration of galectin-1. So in general, the trophoblasts express very high levels of galectin-1. Progesterone, which is the hormone that is very important in the first trimester of the placenta, increases the levels of galectin-1 in the trophoblast, and there is a hierarchical cascade of immunosuppressive events that silence the immune response in the fetus. So galectin-1 is not only important for the tumor to evade the immune response, but also for the placenta to try to keep at bay the fetus that is growing. So one of the findings, one of the observations that we found during the time was that galectin-1 was able to skew the balance of the immune response toward a Th2 cytokine profile in different models of autoimmune disease. And also, galectin-1 favors a Th2 dominant response, an immunosuppressive response during tumor progression and pregnancy. I am seeing a mochila like mine. Are you sure it's yours? Really? Ah, perfect. Okay, no problem. I am just obsessed with my mochila. So, on the other hand, galectin-1 favors a Th2 dominant response during tumor progression and pregnancy. So we asked, what are the rational mechanisms underlying this effect? And one of the most important questions in the literature is the search for selective immunosuppressive agents. You know, we don't have in the literature selective immunosuppressive agents. Glucocorticoids, for example, can eliminate the good immune response and the bad immune response. So there is no selective effect of immunosuppressants for autoimmune disease and for transplant rejection. So we asked whether galectin-1 glycan interactions can selectively modulate the survival of effector T cells, because it kills T cells, and on the other hand, it induces a bias toward Th2 response. So Marta Toscano from my lab, in 2007, she demonstrated that galectin-1, which is a female in Spanish, that's why here is galectina 1, I didn't translate, selectively eliminates Th1 and Th17 cells. And these are the T cell subsets that cause autoimmune diseases. But it can spare Th2 responses because Th2 cells, and we now know that also regulatory T cells, are covered by alpha-2-6 sialic acid. And so alpha-2-6 sialic acid avoids the recognition of the glycoconjugates for galectin-1. So we found that Th1 and Th17 cells share all the repertoire of glycans that are critical for binding of galectin-1, while Th2 cells are protected from galectin-1 by specific sialylation. Consistent with the role of galectin-1 in eliminating Th1 and Th17 cells, which are pathogenic and are the bad guys, we found that when we eliminated galectin-1 gene, there is an increased severity of autoimmune disease in these mice, an increased T helper 1 and Th17 response. So there is an increased severity of the disease caused by the increased number of these cells that are not dying because galectin-1 is not there. So the last and fourth challenge we faced was to trying to dissect the yin-yang of dendritic cells. So I told you at the very beginning of my talk that dendritic cells can be amplifiers or silencers of the immune response. And this is what we call the yin-yang. So the same cells can amplify and switch on, but also in some cases they can switch off the immune response. So what are the molecular mechanisms that underlie these findings? And what we found was, and Juan Ilarregui from my lab found, is that galectin-1 produced by tumors or inflammatory lesions can bind to glycoconjugates on dendritic cells, and these dendritic cells produce a novel cytokine called IL-27. IL-27 is from the same family as IL-12, which is a pro-inflammatory cytokine, but in contrast to IL-12, is a tolerogenic cytokine that generates regulatory T cells. These regulatory T cells produce very high amounts of IL-10, and these immunoregulatory circuits mediated by galectin-1, IL-27, and IL-10 can suppress autoimmune disease, favor the resolution and remission of autoimmune diseases, it can impair resolution of infection, and promote tumor immune escape and metastasis. So here we close the circle of our studies. Most recently, in a paper that we just resubmitted, we found that this type of circles, of circuits and networks can also occur in the central nervous system. So in the case of the central nervous system, in the presence of TGF-beta which is an immunosuppressive cytokine, astrocytes produce very high levels of galectin-1, and in fact are the only cells that express very high amounts of galectin-1 to generate alternatively activated microglia to silence the microglia which suppresses neurodegeneration. This has been done mainly by Ivan Mascanfroni and Sarah Staroson. So the last question we asked was whether galectin-glycan interactions can influence vascular signaling programs. And this was because when we blocked galectin-1 expression in tumors, we always found that there was a blockade of blood vessels growing in the tumor. So we asked whether galectin-1 can also affect angiogenesis. And also, to make a long story short, what we found is that during hypoxia, galectin-1 is increased. It can interact with specific receptor for BGF, which is BGF receptor 2, is a receptor for angiogenesis, and it can promote abnormal angiogenesis in the tumors. So this has been done by Diego Crossi from my lab. And finally, in the last part of my talk, I would like to discuss whether we can capitalize on all this work, on all this basic work, on galectin-1 glycan interactions for therapeutic purposes. And as you know, it's a very long road, but we are starting to try to apply this very basic research for many years for therapeutic purposes. So in the case of cancer, it is important to block galectin-1, because as you remember, galectin-1 killed T cells, and we don't need that galectin-1 will kill T cells. So we need to eliminate galectin-1 from the tumor microenvironment. Also, we found that galectin-1 induces angiogenesis. So blockade of galectin-1 will also reduce angiogenesis. So first, in 2004, we started with the design of, together with the collaboration from Organic Chemistry's department, we started with the design of some synthetic lactulosamine, which are inhibitors of galectin-glycan interactions. But the problem was that it blocks galectin-1, but it also can block other members of the galectin family with a similar glycan specificity. So 3 years ago, we started with the development of monoclonal antibodies with blocking activity. We came to the very old idea of generating monoclonal antibodies which are specific and selective for galectin-1. So after we generated different monoclonal antibodies, some of them cross-react with other galectins, some not, and only one clone had neutralizing and blocking activity. So we are trying to expand and develop this clone. It blocks the carbohydrate recognition domain, and as you can see here, it's a typical model of melanoma in when we inject melanoma cells, tumor cells into mice, and when the tumor volume is 100 millimeters cubic, we inject different doses of the anti-galectin-1 FG-G7 antibody, or its isotype control. As you can see here, there was a significant reduction of tumor volume and also in melanoma, in Kaposi sarcoma, another tumor. And it was dependent on the immune system because when we injected this antibody in an immunodeficient mice, the tumor started to grow. As you can see here in this photograph, injection of the antibody resulted in a reduction of tumor vessels, normalization of the tumor vasculature, and increased levels of Th1 and Th17 responses, which are very important to eliminate the tumor. So not only targets the immune response, but also targets angiogenesis, blood vessels. So for immunotherapy in autoimmune diseases, as you know, injection of galectin-1, administration of galectin-1 can suppress disease in several autoimmune diseases. But what are the obstacles of using recombinant galectin-1 for treating autoimmunity? High concentrations are required to skew the balance of galectin-1 toward the dimeric form, low avidity for N and O glycans, and sensitivity, high sensitivity for inactivation by low pH and oxidation conditions typical of inflammatory microenvironments. So we cannot use, or we have to inject every hour galectin-1 because it gets oxidized, and at the low pH it gets deactivated. So Santiago Dilela from our group is trying to develop what we call a super GAL1. This super GAL1 will have different mutations that will improve lectin activity, dimerization equilibrium, and resistance to pH and oxidation for therapy of autoimmune diseases. So, gathering the pieces of the puzzle together during hypoxia or under the influence of different transcription factors, galectin-1 is secreted from tumors. It can induce apoptosis of T cells, particularly Th1 and Th17 cells, although Th2 cells and Treg cells are protected from galectin-1. It can also initiate a silencing of tolerogenic circuits on dendritic cells, and it can also induce abnormal angiogenesis, mimicking the growth factor VEGF. So, we usually go to different talks when we hear the sentence from bench to bedside or from genes to clinics. So I would like now to highlight from the chicken retina to the therapy of cancer and autoimmune diseases, and this is what we are trying to put all our efforts to try to bring some of our efforts during the past years to the clinics. So, as take-home messages, galectin-1 glycan interactions can contribute to create immunosuppressive microenvironments in tumor and during pregnancy. Placenta protects the growing fetus from the deleterious effects of galectin-1, of T cells via secretion of galectin-1. Differential glycosylation of Th1, Th2, and Th17 cell subsets selectively regulates susceptibility to cell death. Galectin-1 glycan interactions can trigger regulatory circuits on dendritic cells through the novel cytokine IL-27. Astrocytes deactivate microglia and suppress autoimmune inflammation through galectin-glycan interactions. And antibody-mediated galectin-1 blockade promotes tumor remodeling and alleviation of tumor hypoxia and influx of immune effector cells. So finally, using all these elements, what we are trying to do now is to establish a glycomics platform in our lab to try to address some questions related to glycobiology, some basic questions, some clinical questions related to prognosis or diagnosis using different glycoforms, and some questions related to therapy. And the person in charge in our lab is Karina Mariño, who will be part in IBIME and part in the Faculty of Exact and Natural Sciences here. So these are the people from my lab. We are, as you know, we are a lot of people, 22 now, and there are several research investigators. There are several fellows and some technicians. So many people working both in the Laboratorio de Inmunopathologia at IBIME CONICET, and here at the Laboratorio de Glicómica in the Department of Biological Chemistry in this faculty. So I would like to thank all the funding that we had in the past years, my mentors, always Clelia Riera and Carlos Landa from Córdoba, and all our collaborators, many of them from the Faculty of Exact and Natural Sciences, Darío Estrin, here from the that are helping us with all the biochemistry and biophysics of galectins, Julio Caramelo also, and Elba Vázquez from the Department of Biological Chemistry. So I would like also to invite our guest to visit other sites from Argentina apart from Buenos Aires. And thank you very much for your attention.